whole, I'll speak about our motivation for implementing modular approach and the goals we wanted to achieve. Then I'm going to make a brief review of the existing works in this direction and describe our conception of the modular model. The key feature here is application of the visual modeling approach. It requires definition of graphical notation and the algorithm for numerical model generation, appropriate for further simulation. Here we present two different approaches: model flattening and simulation on the basis of agent modeling principles. This approach can also be used in combination. And at last, I'll give examples of approach implementation in complex modular systems modeling. Our modeling of complex biological systems is not a trivial task because comprehensive models are often rather difficult to understand, further support and work with. However, biological systems can be decomposed into functional subsystems which can be modeled independently by different researchers. They can be further aggregated into one model. Models can be validated, improved independently, and replaced by more advanced models. On the other hand, there is a huge number of models describing different biological subsystems created probably by different scales, by different formalisms. Integration of such models is also an interesting problem. However, there is so far lack of specialized tool for convenient visual representation and numerical calculations of model of biological systems. Let's discuss some issues regarding biological system representation. Yuri Lazernik, in his well-known article published in 2002, state a question whether biologists can fix a radio. So here we can see biologist view on radio and engineer view. And Yuri Lazernik is quite pessimistic about biologists' capabilities because using such representation lacks of quantitative information and cannot be used for efficient modeling. In the conclusion to his article, he predicts creation of standards for formalization of biological systems. And now we have some such standards. For example, most known as SBMS, and for formal description of numeric biological model, and SVGN for formal description of its graphical notation. Together they give convenient visual representation of model and strict mathematical foundations of it. And here we can see SVGN model of TNF alpha model. And this representation is maybe compared to engineer view of the problem. However, this approach is far behind modern standards in technical engineering. Moreover, SVGN notation, for example, has several limitations in representation of biophysical processes in system. Here is the example of overall circulation model created by Professor Guyton in 1972. As you can see, it's quite complicated, and even if I'll zoom, it's hard to improve it or reuse, especially for biologists. It was recreated in MATLAB Simulink in 2010. However, we can see it still is complicated. Above leads us to the modern modular design approach, where modules are biological models appropriately combined into one model. So our aims are implementation of modular approach to modeling of complex biological systems, developing of a tool for visual modeling, and algorithm for model numerical simulations. I thought there is no specialized comprehensive tool at this moment. I'll give a short review on works in this direction. A group of scientists from Virginia Polytechnic Institute under the supervision of Professor Tyson proposed 4 approaches to modular modeling. They are fusion, composition, aggregation, and flattening. They propose extension of SBML to include modular modeling in it. Fusion, suppose we have 3 models which we would like to integrate into one model. Fusion means manual creation of a new model on the basis of some models. composition implies integration all information about submodels into one view and direct detailed information about how the elements should interact. Aggregation approach means that modules predefine the interfaces through which they can be connected, and all information about the inner implementation is hidden. And model flattening means automatic fusion procedure on the basis of a model created by either composition or aggregation approaches. A group of researchers from Rennes University, working within the premise of Sapphire project, have formulated two approaches to model modeling in the case of different modules formalisms. As I said before, previous work deals with SBML models. Two approaches are formalist automation, where all modules are transformed to one formalism and further simulated as one model, and co-simulation, which suggests that all modules are simulated independently and interact somehow. Here we can see a picture from the paper. So modular model is composed is hierarchic structure, and to each atomic model corresponds some simulator. They implement co-simulation approach. Now I'll give our conception of modular model. Models, as I said before, are models of biological systems, and they should explicitly specify their interfaces. So module is generally speaking, is some black box which defines its interfaces. Interface means parameter specification. It could be shared parameter, input parameter to the module, and output parameter of the module. If we have two models, we can connect them through undirected connection between two shared parameters, or direct connection. That means that one module passes parameter value to another. A simple example of this approach is given here. Suppose we have three modules, each with one reaction, and here reactant of one reaction is connected with product of another, and that means stitching of those reactions and those species. Direct connection means using this parameter as a modifier for a reaction. For convenience of creating and working with modular models, we utilize the concept of visual modeling. So I need to tell a few words about visual modeling concept. Visual modeling implies three main components. First, it's graphical notation. For example, it's SVGN. Then, numerical model, which is created on the basis of SVGN. For example, in BioMed, we can create a numerical model in Java code on the basis of SVGN model. And simulation result, finally. So, first step is to define our graphical notation. Here it is presented. It includes visual symbols for modules. This is mathematical model containing mathematical model, special technical model for moving average calculation, model followed with simulation relevant chart, model containing one constant value, graphic notation for input port, output port, shared port, directed connection, undirected connection, and a variable on model. It should be noted that there may be several variable elements on the model which are associated with one variable. So we reduce the number of intersection of connections. Here is an example of module called arterial system. Here we can see input parameters. They goes to some differential equations, and they produce output parameters. Red arrows means that parameter calculated in this equation plays positive role in calculating of this parameter, and blue arrows means negative role. Here is another view mode in BioMed. Here equations are hidden, and we just see relations between parameters. Those are physical parameters of the human arterial system. Here is the user interface for model creation in BioMed. Here we can see a list of modules know that some of their parameters means equal biological entities, we may connect them with appropriate connections. And so, continuing this iterative procedure, we add new models and create a large modular model describing the human cardiovascular system. Of course, there are some limitations on connections. For example, we cannot have cycles for one parameter of any type, and we cannot have several incoming connections to one input port. It's clear that those situations do not make sense. We do not know which of these parameters will be used in this module. So we created our model, but for now it's just blue modules connected with some errors. So we need a formal interpretation of this model to obtain numerical model and simulate it. For this we propose two approaches. First is model flattening. It can be used when all modules utilize the same formula, namely differential algebraic equations with events. Models can be SBML SVGN model imported in BioML or model created in BioML with our own notation. And here you can see model containing interconnected models is automatically processed into a list of OD equations, which is flat model in our notation, which can further be simulated in BioML. Next approach is what implemented for general case when models utilize different formulas. We use approach on the basis of agent modeling principles. Agent modeling is widely used in the cases when formal description of system behavior as a whole is too complicated or even impossible. For example, social activities or contagious disease spread. It assumes that the model behavior is a result of activity of several or large number of interacting agents. Basically, agent is some black box which receives a signal and sends a response. So interacting in environment, agents produce system behavior. In our concept, each agent corresponds to module and contains mechanism for functioning in time. For example, if model contains OD system, the agent contains corresponding numerical simulator, and functioning in time means OD system solving. The main part of agent model is a scheduler which synchronizes agents functioning in time and provides correct signal exchange between them. So simulation continues iteratively, and mathematical submodels at the end and the start of each step send parameter changing to the scheduler. Let's conclude. We have two different approaches. In the case of equal formulae, namely die with events, and different formulae. They both have certain advantages and disadvantages. Model flattening is based on strict mathematical foundation as we simulate simply OD die model with events. However, only die models are allowed. In its turn, agent-based simulation has lots of mathematical strictness. However, it does not impose any restrictions on modules formalisms. Only restriction is model type implemented in BioML. In order to compensate the disadvantages, we can use approaches in combinations. Suppose we have model where some modules utilizes die with events formalism and some not. We first apply flattening algorithm and then agent-based simulation. And now I'll brief Second is the iterative nature of model construction. We start from some model and iteratively add a new part or remove to it. At some point, probably we will meet situation when some models are not more needed and will be removed. And so this iterative procedure continues while we obtain our result. Thank you for your